Welcome to The Explainer. Today, we're looking at a story that completely rewrote the rulebook of modern medicine. We are exploring the development of Imatinib. You might know it commercially as Gleevec or Gleevec. And what makes Imatinib so incredibly special is that it serves as the ultimate proof of a concept that, frankly, a lot of scientists thought was pure fantasy. It proved that cancer can actually be treated with pure, calculated logic, rather than just blind poison. It was a problem that seemed downright impossible at the time, but by the end of this, you're going to see exactly how this single molecule decoded that impossible problem and changed literally everything we know about cancer drugs.
Okay, let's dive into this. Here is our roadmap. First, the carpet bombing era. Second, the Philadelphia chromosome mystery. Third, designing the perfect doorstop. Fourth, a clinical miracle. And finally, we'll wrap up with the new oncology blueprint.
Section 1. The Carpet Bombing Era. Oncology before the 1990s.
So to truly appreciate this breakthrough, we really have to establish exactly what scientists, and more importantly, patients, were up against in the last century. Up until the 1990s, cancer chemotherapy was, well, honestly, it was essentially a strategy of carpet bombing the human body. Drugs like plitin, doxorubicin, and cyclophosphamide were terrifyingly blunt instruments. They worked on a pretty basic, albeit brutal, premise. Cancer cells divide faster than most normal cells, right? So the idea was, if you introduce a systemic poison that targets dividing cells, the cancer will theoretically die just a little bit faster than the patient does. But we have to look at the massive downside here. It attacks all fast-dividing cells without any prejudice. The medicine just cannot tell the difference between a rapidly dividing tumor cell and, say, a healthy hair follicle or the lining of your stomach or your bone marrow. And this is exactly why traditional chemotherapy patients suffer from hair loss, severe gut damage, and devastating immune system collapse. Those healthy tissues naturally divide fast too, so they just become tragic collateral damage.
Oncology desperately needed a paradigm shift. I mean, we really needed to move away from this toxic scattershot approach. The burning question was, what if, instead of destroying everything in a toxic cloud, we could prove that some cancers are actually addicted to a single abnormal signal? What if we could find that exact bullseye and just turn it off?
Section 2. The Philadelphia chromosome mystery, a decades-long detective story.
This brings us to a specific fatal blood cancer called chronic myeloid leukemia, or CML, and a medical mystery that started decades before the drug was even a thought. Let's walk through this timeline together. Back in 1960, two researchers in Philadelphia, Peter Nowell and David Hungerford, noticed something really odd under the microscope. Patients with CML had this weirdly short chromosome 22. They dubbed it the Philadelphia chromosome. By the 1980s, science had advanced enough to realize this was actually a translocation. To understand a translocation, imagine your DNA is a massive set of encyclopedias. What happened here is that a page from volume 9 was ripped out and glued right into the middle of volume 22. This was a huge deal because chromosome 9 contains ABL, which is an enzyme known as a tyrosine kinase that tells cells to grow and divide. Chromosome 22, on the other hand, contains a gene called BCR. Fast forward to the 1990s, and biochemist Nicholas Leiden at Ciba-Geigy, which, by the way, later became Novartis, and an obsessed oncologist in Oregon named Brian Drucker made a massive bet. They decided they were going to target this fused, mutated gene, even though the broader pharmaceutical industry was basically telling them it was impossible.
Now, what's really interesting about this is what happens when those two chromosomes swap. ABL and BCR fuse to create BCR-ABL. It's essentially a Frankenstein protein. And why a Frankenstein? Because it's two completely unrelated pieces of genetic code forced together, creating this monster that just doesn't obey normal cellular rules anymore. Normally, a kinase like ABL needs external signals to tell a cell to grow, but BCR-ABL is stuck in the on position all the time. It never waits for a signal, it just fires constantly. So CML in a single sentence is basically this: white blood cells carrying this mutant BCR-ABL protein are told to never ever stop dividing, which ends up filling the bloodstream with useless cancerous cells.
Section 3. Designing the perfect doorstop. Rational drug design.
So, if Leiden and Drucker were right, blocking this specific protein would cure the cancer. But how do you actually build a drug to do that? Well, kinases are basically cellular machines, and they work by taking ATP, which is cellular energy, binding it in a tiny little pocket, and transferring its phosphate to a target protein to send a growth signal. Think of that ATP pocket like the tiny ignition switch on a car. The logic was actually incredibly simple. Block the ATP pocket, and you block the kinase from ever turning on. To do this, chemists Jürg Zimmermann and Elisabeth Buchdanger synthesized a huge library of compounds called phenylaminopyrimidines and meticulously screened them.
Their lead compound was STI-571, which we now know as Imatinib, and crystallogically later revealed something amazing. Imatinib doesn't just jam itself into the active form of the enzyme. Instead, it fits perfectly into the inactive, closed conformation of ABL. It slides right into the ATP pocket and wedges itself into an adjacent hydrophobic pocket that only exists when the kinase is turned off. It acts quite literally like snapping a key off in the ignition so the machine can never be started again. It is, for all intents and purposes, a perfect molecular doorstop.
Now, at the time, Big Pharma thought kinase inhibitors would be incredibly toxic. I mean, there were over 500 different kinases in the human body, and they all use ATP for energy. The fear was that if your drug wasn't specific, it would shut down every single kinase in the body, which would be instantly fatal. So how do you block just one? Well, while Imatinib isn't perfectly specific, it does hit a couple of others like c-KIT and PDGFR, it is specific enough. It proved to be 100 times more selective for BCR-ABL than most other kinases. And that 100 times selectivity is exactly what separates a miraculous, life-saving drug from a lethal poison. You could hit the target without taking out the whole system.
Section 4. A clinical miracle. The Phase 1 and 2 trials.
Okay, so we have the lock and we have the newly designed key. The next step was taking this into the clinic, and the emotional payoff here is just staggering. In 1998, Brian Drucker ran a Phase 1 trial in Oregon. The patients in this trial were completely out of options. I mean, they were desperate. They had failed every treatment available, including heavy, painful regimens of interferon, and they were facing a really grim reality of dying within 3 to 5 years. Out of the 54 patients in that trial, 53 achieved a complete hematologic remission. Let that sink in for a second. 53 out of 54. Within weeks, their blood went from being densely packed with cancerous white cells to looking exactly like the blood of a perfectly healthy person. And that 54th patient, they actually had a different mutation altogether. But best of all, there was almost zero chemo-like toxicity, none of those carpet-bombing side effects.
The success was so undeniable and so rapid that the medical community was just utterly stunned. The subsequent Phase 2 data in 2001 actually landed on the cover of Time magazine. Because the results were so clearly saving lives in real time, the FDA approved Imatinib in a record-shattering 2.5 months in May of 2001. A 2.5-month approval timeline is practically unheard of in pharmaceutical development. It was just an astonishing validation of the science. And this brilliantly illustrates the real-world impact of that approval. Before Imatinib, the 5-year survival rate for chronic myeloid leukemia was around 30%. The only potential cure was a brutal bone marrow transplant, which carried a 20 to 30% mortality rate all on its own. After Imatinib, the 10-year survival rate skyrocketed to nearly 90%. Take a second to just let that jump from 30% to 90% truly sink in. CML transformed practically overnight from a death sentence into a manageable chronic disease. Today, a patient takes just one 400 milligram pill a day and can live a near-normal lifespan.
Section 5. The new oncology blueprint. The legacy of Imatinib.
Imatinib wasn't just a miraculous cure for one rare leukemia, though. It actually became the skeleton key that unlocked an entirely new era of medicine. The legacy of this drug can be broken down into 3 massive impacts. First, it definitively proved the concept of oncogene addiction, the idea that some cancers truly live and die based on a single driver mutation. Second, it created the literal blueprint for targeted therapy. Follow the Linden-Drucker logic. Find the driver, make a key for its lock. This birthed an entire generation of targeted drugs: erlotinib for EGFR mutant lung cancer, trastuzumab for HER2 breast cancer, and vemurafenib for BRAF melanoma. Third, it forced us to solve the problem of drug resistance. See, about 20 to 30% of CML patients eventually develop resistance because the cancer mutates the physical shape of the protein's pocket. It's often a mutation called T315I. Think of it like a lock mutating its internal pins so the old key just doesn't fit anymore. But because we understood the exact mechanism, scientists designed second and third generation inhibitors, drugs like dasatinib, nilotinib, and ponatinib, that act as master keys, binding to the mutated shapes and beating the resistance.
Another incredible legacy of Imatinib is how we track cancer. Today, we don't just look at simple white blood cell counts under a microscope. We use PCR molecular monitoring. To explain this simply, instead of just looking at the overall shape of a haystack to see if it looks wrong, PCR lets us search for the exact genetic needle in that haystack at the DNA level, finding microscopic traces of the BCR-ABL transcript in the blood. This entire concept of highly sensitive molecular monitoring started right here with Imatinib. Now, it's worth noting that while it is remarkably well tolerated compared to classic chemo, it is still a powerful drug. It can cause mild periorbital edema, muscle cramps, or nausea, and it interacts with the CYP3A4 enzyme, meaning long-term cardiac and liver health still requires careful tracking by a specialist. But compared to the carpet bombing era, we are in a completely different universe.
Which brings us to the ultimate thesis of our explainer today. Nicholas Ludden and Brian Drucker looked at a fatal leukemia, traced it down to a single broken enzyme caused by a single ripped-out chromosome page, and painstakingly built a specific molecule to shut down only that enzyme. It was the absolute first time in human history we cured a cancer with precise logic instead of blind poison.
So, as we wrap up, I want to leave you with this thought. If a single swapped chromosome in a fatal leukemia could be decoded, neutralized, and managed with a daily pill, what other impossible medical locks are hiding in the human body, just waiting for the right molecular key to be designed? The dawn of precision oncology started with Imatinib, but the rest of the day is just getting started. Thank you so much for joining me for this explainer. Keep asking questions and keep exploring the incredible logic of science.